Okay, Assalamualaikum and a very good morning to Dr. Liziana. I'm Hidayah from Group 8 and I will be presenting about Practical Lab 4, which is the digestion of starch and lipid. Okay, but first I'm going to introduce our group members. First we have Hidayah, which is me. Second, Alia, Hadi Husni and also Alia Maisara. Okay, next I'm going to talk about the objective of our experiment. Our first objective is to examine the function of common enzyme in digestion of starch and lipid. Second, we want to determine the factors affecting the enzyme activity.
Okay, welcome to methodology slide. So we are going to review the methods of starch experiment. So what we need to do for this experiment is we are going to prepare 5 pairs of test tubes as follows. Label A, B, C, D, and E. And each of the label we need to pair it with 2 test tube. So as we can see from the table, test tube A, we have A1 and A2. B, B1, B2, C, C1, C2, D, D1, B2, E, D1 and E2. And each of the label we need to put different content. As example, label A we need put 4 ml glucose only. For B, we are going to put 5 ml starch only. C, 4 ml untreated amylase only. D, 2 ml starch and 2 ml untreated amylase. And E, 2 ml starch plus 2 ml acidified amylase. Okay, after that, the second method is we need to shake well all the test tube and leave it in a stand condition for 10 minutes. After that, add the test reagent as follows. Add 0.5 ml of iodine solution into test tube A1 until E1. It means that A1, B1, C1, D1 and E1. Shake well by using vortex and record your observation. Next, add 0.5 ml of Benedict's reagent into test tubes A2 until E2. Shake well and incubate in boiling water bath 100 Celsius for 5 minutes. Record your observation, and then after that wait for 10 minutes after we incubate it and record your observation. And after that, complete the table below.
In digestion of starch by amylase experiment, we use 5 pair of test tube named A, B, C, D and E, each with its content, and all either go iodine test or Benedict test. In test tube pair A, we put 4 ml of glucose and each of them undergoes iodine test and Benedict test. The iodine test show no reaction while Benedict test form a brick red precipitate, concluding that glucose is present while starch is absent. In test tube pair B, only 4 ml of starch is placed into it, and iodine turns blue-black while Benedict solution doesn't react. It shows that glucose is absent while starch is present. In test tube pair C, 4 ml of untreated amylase is placed inside the test tube, and iodine tests show no reaction while Benedict tests show result in orange-red precipitate. This shows that glucose is present while starch is absent. In test tube pair D, 2 ml of starch and 4 ml of untreated amylase is placed inside of the test tube. The iodine test is the iodine turns blue-black while the Benedict test form an orange-red precipitate. This shows that glucose and starch both is present. In test tube pair D, 2 ml of starch and 4 ml of acidified amylase is placed inside of the test tube. Iodine turns blue-black and Benedict solution make a orange-red precipitate. This shows that both glucose and starch is present.
Welcome to another slide of methodology for another experiment, which is lipid experiment. So what we need to do for this experiment is first prepare test tubes as follows, and we require 4 test tubes, and we label it as A, B, C and D. Each of the test tube contains different content and different condition. So as we can see from the table, test tube A, we need to put 2 ml of lipase and 2 ml of bile. Then add 2 until 3 drops of phenolphthalein and 0.2% sodium hydroxide drop by drop until the solution turns pink. Add 3 drops of vegetable oil and shake well. After that, incubate at 37 Celsius for 1 hour in water bath. Next, for the test tube B, we need to add 2 ml of lipase and 4 ml of distilled water. Then, add 2 to 3 drops phenolphthalein and 0.2% of sodium hydroxide drop by drop until the solution turns pink. Then, add 3 drops of vegetable oil and shake well. Then, incubate for 37 Celsius for 1 hour too. Okay, for the C, we construct our own control for role of lipase, and D, construct our own control for the effect of temperature. For next, observe the initial color and the final color of each test tube and record it in table 4.3.
In digestion of lipid by lipase experiment, we use 4 test tubes and observe the initial and final observation after an hour. The test tube A has 2 ml of lipase and 4 ml of bile and is placed in 37 degrees Celsius. It showed no changes, thus making a negative result. In test tube B, 2 ml of lipase and 4 ml of distilled water in 37 degrees Celsius is put into the water bath, and the initial and final observation doesn't change. This also shows negative result. In test tube C, however, 4 ml of bile and 2 ml of distilled water is put inside of a 37 degree water bath. Initial observation and final observation is the same, thus showing negative result. While D have 2 ml of lipase and 4 ml of bile put inside a room temperature, it is shown that initial observation has a more rich color than the final observation. This shows that the digestion occurs, thus positive result has been obtained.
Okay, hi again. So now I'm going to discuss about the digestion of starch. Okay, for the first pair, test tubes A, we get positive result because the blue color of Benedict solution turns orange red precipitate, which indicates there is glucose in the test tubes. Okay, for the second pair, we also get the same result, which is positive because in test tube B, there's contain starch. So the brown color of the iodine solution turns blue-black, which means there's starch in the test tubes. Okay, but for the C test tubes, which is the third pair, sadly we got negative result because the blue color of the Benedict solution turns orange red precipitate, which means there is glucose in the solution. But in test tube C, we put untreated amylase, which do not contain glucose. So what can we do to fix this problem? We can rinse the test tube first, which we use distilled water to make sure there is no excess chemicals in the test tubes before doing the experiment. Okay, for the fourth pair, test tubes D, we get the positive result. This is because the brown color of iodine solution turns blue-black, and also the blue color of Benedict solution turns orange red precipitates. In these test tubes, we put untreated amylase and also starch. This means the amylase break down the starch into glucose, so there's glucose present. Okay, lastly, in the last pair of the test tubes, we put starch and also acidified amylase. So the same thing happened. The amylase break down the starch into glucose. So there's 2 starch and glucose present, which means the blue color of Benedict solution turns orange red precipitate and the brown color of iodine solution turns blue-black.
Okay, we are going to continue our presentation with the discussion on the lipid digestion experiment. So, in this experiment, we have prepared 4 test tubes, which are test tube A, test tube B, test tube C, and test tube D. So, for test tube A, the result obtained is negative result The error in this part of experiment is the result obtained should have been positive as it is in optimum condition. So we have figured out the reason behind this error is because an excessive amount of sodium hydroxide was added into the solution. It causes the solution to not to be an optimum condition, which is in high alkaline state instead of the pH 8. So how to fix this error is we have to add the sodium hydroxide moderately and immediately stop when the color of the solution turns pink. So for the second test tube, which is test tube B, the result obtained is negative. The error in this experiment is the result obtained should have been positive. So the reason behind this error is because an excessive amount of sodium hydroxide was added into the solution. It causes the solution not to be in optimum condition, which is in a high alkaline state. So how we fix this problem? By adding the sodium hydroxide solution moderately and immediately stop when the color of the solution turns pink. For test tube C, the result obtained is negative result. In this part of experiment, there is no error reported. This is because the result obtained was the same as the expected result. The reason behind this result is because there is no digestion of lipid occurred. This is due to the absence of lipids in the test tube. For test tube D, the result obtained is positive. There is no error reported in this part of experiment because the result obtained was the same as expected result. So the reason is that a process of digestion lipid happened, but the digestion lipid occurred in a lower rate because the solution was not in optimum temperature, which is 37 Celsius. That is all from us. Thank you.